let us review some key terms on biotechnology. Anneal. In molecular biology, the process by which two single strands of DNA hydrogen bond at complementary nucleotides to form a double-stranded molecule. Biomarker. An individual protein that is uniquely produced in a diseased state. Biotechnology. The use of artificial methods to modify the genetic material of living organisms or cells to produce novel compounds or to perform new functions. Cloning. The production of an exact copy, specifically an exact genetic copy, of a gene, cell, or organism. Gel electrophoresis. A technique used to separate molecules on the basis of their ability to migrate through a semi-solid gel in response to an electric current. Gene therapy. The technique used to cure heritable diseases by replacing mutant genes with good genes. Genetic engineering. Alteration of the genetic makeup of an organism using the molecular methods of biotechnology.
Hi, if you found this video informative, give it a thumbs up, subscribe, and share the link. We want these free textbook resources to be available to other students. Alright, let's get back to reading.
Genetic map. An outline of genes and their location on a chromosome that is based on recombination frequencies between markers. Genetic testing. Identifying gene variants in an individual that may lead to a genetic disease in that individual. Genetically modified organisms, GMO. An organism whose genome has been artificially changed. Genomics. The study of entire genomes, including the complete set of genes, their nucleotide sequence and organization, and their interactions within a species and with other species. Metagenomics. The study of the collective genomes of multiple species that grow and interact in an environmental niche. Model organism. A species that is studied and used as a model to understand the biological processes in other species represented by the model organism. Pharmacogenomics. The study of drug interactions with the genome or proteome, also called toxicogenomics. Physical map. A representation of the physical distance between genes or genetic markers. Plasmid. A small circular molecule of DNA found in bacteria that replicates independently of the main bacterial chromosome. Plasmids code for some important traits for bacteria and can be used as vectors to transport DNA into bacteria in genetic engineering applications. Polymerase chain reaction, or PCR, a technique used to make multiple copies of DNA. Protein signature, a set of over- and underexpressed proteins characteristic of cells in a particular diseased tissue. Proteomics, study of the function of proteomes. Recombinant DNA, a combination of DNA fragments generated by molecular cloning that does not exist in nature. Recombinant protein, a protein that is expressed from recombinant DNA molecules. Reproductive cloning, cloning of entire organisms. Restriction enzyme, an enzyme that recognizes a specific nucleotide sequence in DNA and cuts the DNA double strand at the recognition site, often with a staggered cut leaving short single strands or sticky ends. Reverse genetics, a form of genetic analysis that manipulates DNA to disrupt or affect the product of a gene to analyze the gene's function. Transgenic, describing an organism that receives DNA from a different species. Whole genome sequencing, a process that determines the nucleotide sequence of an entire genome. Now on to chapter summary.